Hello, so this is a video of card test. Test.
Hey,
Hey,
what's up?
what's up?
Not much.
I got your test.
Yeah. That's the answer key.
Mm-hmm. So, which ones did you have trouble on?
Um, like stuff like number 5.
Alright, let's see. You treat a partially purified preparation of protein with a reagent that breaks bonds between sulfur atoms. Which level? Okay, so the bonds between sulfur atoms, they're called disulfide bonds. And even if you didn't really know that, you can still solve it based on what you know about, well, primary structure is what type of bond.
That's an amino acid.
It's when amino acids polymerize, right?
Yeah.
Can you tell me what it is, like, chemically speaking, like the bond, like what attaches to what?
I don't know, hydrogen bond?
No. So it's basically, well, okay, it's basically the hydrogen of the amine group of one amino acid, which attaches to the OH group of the carboxyl of the other amino acid, forming that peptide bond. So it seems like they are putting an emphasis on the biochem aspect of it, so I'm going to, like, you know, focus on the biochem part from now on, in addition to everything else, of course. Because so far I was just kind of glossing over it. But here, do you see, oh wait, let me share my screen. Okay. Check out my screen. See this over here? So this over here, do you see it? Okay, do you see my screen?
Yeah.
Alright, so this over here is, and you should like draw this so you start to get used to what the structure looks like. But this is a generic structure for an amino acid. Now, you have the N terminal, which is the amine group, so it's an NH2 over here.
Mm-hmm.
And then you have the C terminal, which is the carboxylic acid. So it's a carbon that has a double bonded oxygen and an OH group. And this R is just the side chain. So that is what determines how one amino acid is different from another amino acid.
Okay.
All right. Now, here we have two different amino acids, right? And they're going to form a peptide bond linking the two into a dipeptide. So what essentially happens is, you see how a hydrogen from the amine group of one amino acid and the OH group of the carboxylic acid of the other amino acid, that forms an H2O, which leaves through dehydration synthesis. So now there's a bond that's made between this nitrogen and this, like, essentially like OH group, but the OH group leaves. So you have this over here. This is the dipeptide over here. So you have the first amino acid here and the second one here, and they're linked here. That's the peptide bond over there. So it's good for you to draw it out so you get used to it. So you get used to, you know, what the structure looks like. But in a primary structure, you're just going to see this type of bond. All right?
Okay.
Now, in a secondary structure, that is where you only see hydrogen bonds.
Okay.
So, in tertiary and quaternary structures, you have interactions between the side chain groups, like the R groups. So for number 5, if they're talking about a bond between sulfur atoms, right? It's not a peptide bond and it's not a hydrogen bond, which rules out primary structure and secondary structure. So therefore, it must be, through process of elimination, tertiary and quaternary. Okay? So yeah, just, you know, take the time to, like, draw this out so you're used to seeing and drawing out what an amino acid, you know, looks like, what the structure of it is, because that's very important for understanding the type of bonds that, you know, it undergoes in the various different structures, like primary through quaternary. All right, any questions there?
No.
Okay. All right, which is the next one? The next one that you had trouble with?
Mhm. Oh, 37.
How is active IRP, iron regulatory protein, used to regulate gene expression? Oh yeah, we didn't even talk about this. It wasn't even on any of the slides. So I guess none of the other answers is correct. That's pretty weird. Here, let me look this up. Iron regulatory protein. It seems like it's just involved in iron metabolism and iron transport. So that could be just like a trick question where it's none of this stuff.
Yeah.
So I was actually looking into the different types of, because I mean, I knew for a long time, like over 6 months, that it's good to mix the COVID vaccines. And a few days ago, the FDA finally approved it, and yeah, when I was looking into it, I realized it's a good kind of learning opportunity for you because it involves like exactly what we're covering in this course. So what do you know about the different types of the vaccines?
Well, they're all mRNA.
So there's the mRNA one, which is brand new, right? It's never been done before. Why is that so new? Like, why why haven't they done that before?
Well, because it's using your basically genetic code in order to fight against it.
Well, why not use DNA then? If DNA is the genetic code, right?
Well, mRNA makes like proteins that can fight against certain things, or make certain things.
Yeah, but isn't mRNA from DNA?
Wait, say that again?
Well, so mRNA is from DNA, isn't it? mRNA is simply something that is transcribed from DNA, right?
Yeah.
So, why is it so hard to use the mRNA? Like, why weren't they able to do it before? What is something that you should know about mRNA?
I don't know.
Think about its stability relative to DNA.
I see. Okay.
So, what do you see?
It'll probably be able to hold against or fight off of foreign substance.
All right. So check this out. So basically, what we're doing is, so a vaccine traditionally is supposed to be some weakened form of the virus, right?
Yeah.
Because then your immune system has a taste for the virus, so when the actual virus comes in, it has, memory B cells, it has antibodies that can fight that virus, right?
Yeah.
So what they have made are that, so for the mRNA vaccine, they essentially make, it's essentially mRNA that codes for the spike protein in the virus. So it makes that protein, which our immune system recognizes and makes antibodies against. Now, the Johnson & Johnson one, right, that uses DNA rather than mRNA. Now, in terms of stability, what were you going to say?
No, I said
So in terms of stability, right, which is more stable?
All of the mRNA.
Well, mRNA is actually very
DNA.
Yeah, because if you think about it, DNA doesn't change, right? DNA, I mean, like, you get mutate, obviously, but DNA is basically in the nucleus, where it's basically in a vault. But mRNA, right, it is only made when a protein needs to be made. So it's transcribed, like DNA is transcribed into mRNA, and the 5 cap has to be added, the poly A tail has to be added to protect the ends, and it goes into the cytoplasm, and it gets translated, and if it doesn't, it just gets degraded very, very quickly. So remember that mRNA is very, very unstable. Now, DNA, what does that stand for?
What does what stand for?
DNA.
Deoxyribonucleic acid.
Good. And RNA is just ribonucleic acid, right?
Yeah.
So if DNA is deoxy and RNA is just without that, that means that RNA has an extra oxy group, right?
Yeah.
So RNA here. So RNA, right? Here, do you see this this picture over here?
Yeah.
So RNA has an OH group both here and here, right?
Yeah.
So what it does is it can hydrolyze itself. It can break itself apart. Okay? So that's why throughout history they were never able to make an mRNA vaccine, because mRNA is so fragile. So what they did in, the Pfizer and the Mrna mRNA vaccines is they put it in lipid nanoparticles.
Uh-huh.
So they put it in these spheres of lipids where it can enter the cell that way. And that's basically, a brand new thing that's never been done before. Whereas the Johnson & Johnson one is an adenovirus, which is just a vector for taking the DNA portion and putting that into the nucleus. Does that make sense?
Yeah.
Okay. I just want to cover a lot of these other conceptual things, and to get you to think critically, so whatever happens on the next test or whatever, you'll be ready for it, ready for anything really. So yeah, for 37, it wasn't covered in any of the slides, and I'm pretty sure this is a trick question because the answer is, none of the answers are correct. And then I looked it up, and it's just involved in, iron, regulation. So, and if you just go through the choices, A says causes degradation of the mRNA by exonucleases, and B is endonucleases. What's the difference between an exonuclease and an endonuclease?
Exo is outside, endo is inside.
Elaborate. What does that mean exactly?
Don't exonucleases break down from the outside while endos break down from the end?
So from the outside as in the ends of the RNA?
Yeah.
Good. And what does mRNA have that protects it from exonucleases?
Poly-A tail.
Anything else? That only protects one side. What about the other side?
I'm not sure.
5 cap?
Oh, it was that obvious. Okay.
Mm-hmm. But it doesn't have anything to protect it against endonucleases, right?
No.
So that's why it's still going to break down. You basically never have just random mRNA in the cytoplasm. Either it gets used immediately, translated, or it gets broken down.
Yeah.
Okay, let's see. C says it interferes physically with the binding of a ribosome to the 3' end of the ferritin mRNA. Okay, ferritin is, the FER part means iron, And ferritin is just a protein that stores iron.
Okay.
So it looks like this. You see my screen?
Yeah.
Okay. Now, the test says interferes physically with the binding of a ribosome to the 3' end of the ferritin mRNA.
Uh-huh.
So it seems like iron responsive. So iron regulatory protein. This is iron responsive element. Iron regulatory protein. IRP1 and IRP2, they register cytosolic iron concentrations and post-transcriptionally regulate expression of iron metabolism genes to optimize cellular iron availability. So, okay, so that's what that does. They bind to IREs, iron responsive elements, in the untranslated regions of mRNAs for transferrin receptor in both H and L subunits of ferritin. Okay. Iron binding proteins. Alright. So C is really just talking about the ribosome, that, the large and small subunit coming together to cause the mRNA to be translated, and it doesn't really affect that. And then D talks about adding acetyl groups, but that would be, something like an enzyme would do, like an acetylase or something. Do you remember what acetylating does to the histones? Does it make it tighter or more loose?
It makes it loose.
Exactly. So D would be wrong for that reason as well, because it says adds acetyl groups to histones leading to a tight coiling of DNA. So that one would just be through process of elimination.
Okay.
Okay. Which other one did you have trouble with? I mean, you got 19 wrong here, right? Like 19 questions wrong?
No. Hold on, let me see how I graded this.
Okay.
Because I took it as I got an actual 30%.
Yeah, because I did the math, and a 38% would be 19. Like 19 over 50 is 30.38. Oh, wait, so that's 19 correct. My bad. So 31 questions wrong.
Yeah, and I don't know how. But...
Yeah, so let's go over the ones that you got wrong. Because so far you told me, what did you tell me? You told me number 5, and then you told me number 37. but I'm sure there's a lot more in between.
Yeah, the thing is, I'm looking at this and I don't see like I don't see how I got the others wrong unless I just filled in the bubbles wrong.
What does it tell you which ones you got wrong?
No,
No, it doesn't.
no.
Okay. All
Let me
right.
see.
Yeah.
Uh, what? I mean, yeah, I don't know what else to really go over because I don't know what I got wrong and
All right. Let's
everything
just
here.
let's just go over everything then. Because it's worth it because you still have to make sure that this stuff you know it like the back of your hand because if you didn't remember like the 5 cap and stuff like that, it's worth just going over everything. I also opened up the textbook that you sent me like months ago as well, because I think that's also key because when I would be taking these classes in college, I never just looked at the PowerPoints, and that's what we've been doing, just been looking at the PowerPoints. Like I would look at like the actual textbook, just to be completely, to make sure that everything is covered. So I mean, like, yeah, let's look at the first one. The clustering of codons in the genetic code tables provides what advantage to living organisms? What's a codon?
Where are you reading?
It's just the first question on the test. But tell me what a codon is.
It's like like CGA, stuff like that. 3 letters. Like start and stop codons and stuff like that within genetic code.
Is it just start and stop codons?
No.
What else?
Just regular codons.
What do they do?
They keep the sequence going.
All right, but try better, try harder. Of course, like they'll keep it going if but what try to be like more specific. Like, what is a codon? What does it do? What's its function? What happens to it? If you're tired, you can get some coffee or something.
No, I'm tired. I'm just in pain.
Yeah, I of course, like, sorry for that, but I want you to to do well in this.
Yeah. I don't think it helped that I took this test when I went to after I came back from the hospital.
Is there you can't you couldn't have gotten like a doctor's note or something or some type of accommodation?
I would have needed to put that beforehand.
Yeah.
And it wasn't an emergency room visit; it was for an X-ray.
But for your condition, everything is essentially an emergency, because you can't really know in advance how you're going to feel. I think it would be worth it to tell your professor, the entire situation, so he can, take your most recent score into consideration.
Okay.
So like, this is like how I want you to answer. Like, codon is a set of 3 nucleotides on an mRNA that is read in the ribosome by the tRNA, which has a complementary anticodon, which is associated with a particular amino acid.
Yeah.
So, let's say the codon says CAG. The anticodon would say GTC, and that anticodon on the tRNA is going to code for a specific amino acid. Let's say it's like methionine, and that's what it would do. It basically is a thing that connects the the nucleic acid, like the base pairs, to the actual amino acid. So try to, like, I'll ask you these questions and try to give me as much information as possible about what the thing is, just to make sure that we you have it completely known.
Yeah.
Okay, so in addition to start and stop codons, all of the other ones which aren't start and stop codons are codons that code for a specific amino acid, right?
Yeah.
How many amino acids are there?
Isn't it like 23?
20.
20?
And how many codons are there?
Isn't this 63?
64.
64.
How many of those are stop codons?
3.
How many of those are start codons?
Isn't it also 3?
Just one.
Oh, one is the...
So how many of them are codons for amino acids?
61.
Yeah. Oh, I forgot to say that a start codon codes for an amino acid as well. It codes for methionine.
Yeah.
So that's why there's 3 stop codons, 1 start codon, and 64 minus 3 codons for amino acids, because the start codon also codes for an amino acid.
Okay.
How do you get that number, the 64?
Isn't that just combinations of all start and stop codons with every other codon in between?
Yeah, so let's say, so how would you do that? So, like, yeah, how would you get that number? Like, tell me the math of it. So think about it like this. So codon has 3 slots, right?
Yeah.
So in the first slot, how many nucleotides can be there, like in terms of the total nucleotides that exist? In terms of, base, the bases.
Wait, is that one?
Oh, yeah, sorry. Yeah, I didn't really articulate that well. So imagine there's 3 slots, and in that first slot, how many, there's adenosine, how many of those, exist?
4.
All right.
Well,
So,
4.
Yeah, 4. And in the second slot, how many?
Isn't there 4 in each slot?
Good. So it's 4 times 4 times 4. In other words, 4 to the 3rd power. So
Yeah.
what do you get when you do that?
64.
Exactly. Okay?
Yeah.
Good. Now, is that your medication?
That's a lead.
Okay.
I already took my medication earlier.
Mm-hmm.
Let's continue.
Yeah, I'm going to look through the test and the textbook to think of questions to ask you.
I'm gonna get water.
Yeah.
Right. And after this, we gotta go through like 3 slides.
Oh, you got
3
slides?
sets of them. Yeah, I have 3 sets of slides.
All right, yeah. Send.
Yeah, I have a midterm next Wednesday again.
Oh, damn. Okay, yeah. Let's do the slides first because I'm not really sure which ones to go over here, but I do want to like go as I go over the slides. I'm gonna like kind of like ask you like more questions and things like that, just so you have. Yeah. And I'm looking at the textbook also.
All right, send it over. First is membrane structure, then the other 2.
Yeah.
Membrane transport and cytomembrane.
Okay. So I got membrane structure open. You ready?
Uh-huh.
All right. Membranes act as selective barriers. What does a selective barrier mean?
It like takes in only certain things.
Good.
It'll leave certain things out.
Yeah. Okay, so this is plasma membrane is involved in cell communication, import/export of molecules, and cell growth and motility. What's motility?
Like how fast something can move
Just movement, basically.
or movement.
Okay. These are an example of membrane compartments in the eukaryote. So, we have nucleus.
Where are you on? Where are you on?
Oh, do you see my shared screen right here?
No, I don't see your screen.
What?
Oh, wait, now I do. All right,
Okay.
I see it. I got it.
So these are all membrane compartments. And apparently the plasma membrane, it's just 2% of it. But the ER, the endoplasmic reticulum, is 50% of it. So the endoplasmic reticulum is much larger, at least in terms of surface area.
Okay.
So the model that we use for the plasma membrane is called a fluid mosaic model. So we obviously know about the lipid bilayer, but and that's the fluid part of the fluid mosaic, but the mosaic is the proteins that are also there embedded in the plasma membrane. And so, we see that in this picture here. We have peripheral proteins, which pretty much are mostly on the hydrophilic part, like the inside or the outside. We have, so that's this one over here. And then we have hydrophobic alpha helix, which is a transmembrane or integral protein, which means that it goes through the cell membrane. So that's why, because this inner portion of the cell membrane is hydrophobic, right? And therefore the part of the protein that goes through that inner portion must be hydrophobic as well. So these are integral proteins. And on the outer portion, we have attached to the protein oligosaccharides, as well as... Oh, okay. So so what's an oligosaccharide?
And that we'll need to go over again.
Well, what's a saccharide?
It's like a fat almost. Oh no.
What does it sound like?
Saccharide.
In terms of food, what does it sound like? From the options.
Protein?
From the options, lipid, protein, sugar.
Sounds like a sugar.
Yeah. So it's a sugar.
Yeah.
Yep. So oligo just means, a couple. Right? Like there's monosaccharide, disaccharide, polysaccharide. Oligo is just like a couple. It's in between di and poly. So what then is a glycoprotein?
A glycoprotein?
Yeah.
It's a protein that contains glycogen, I'm guessing.
Or just a sugar.
Or sugar.
Yeah. So that's why in these images, right, we have oligosaccharides attached to these proteins, thus making them glycoproteins.
Yeah.
Now, what's a glycolipid?
A lipid that contains sugar.
Exactly. Phospholipid?
A lipid that contains a phosphate group.
Good. What's the difference between a peripheral protein and an integral protein?
A lipid protein, you said?
Oh, no, between a peripheral protein and an integral protein.
Peripheral protein is probably
peripheral?
like a protein that's like
this over here?
So
this is a peripheral protein, and these two are integral proteins. What's the difference?
one is like outside and around, the other one's within.
All right, now tell me what that means in terms of hydrophobic, hydrophilic.
A peripheral protein, maybe a protein that can't get in because it's hydrophobic, so it can't get through the hydrophilic bilayer.
Well, if it can't get through the okay, look at my cursor. Tell me if the region is hydrophobic or hydrophilic. So if the cursor is here, hydrophobic or hydrophilic?
That would be extracellular. Okay, so then that's hydrophilic, yeah.
What do I hear?
Hydrophilic.
Here?
Phobic.
Okay. Peripheral protein, it's just...
Yeah.
So peripheral protein will be what?
Hydrophobic.
Well,
Philic.
good. And then
Sorry,
the integral proteins in these regions would be what?
hydrophobic because
Good.
they can't get through the other one.
So the it's the other way around. So like hydrophilic ones can't get through because this middle portion is hydrophobic, right?
Yeah.
So peripheral proteins, since they're hydrophilic, they can't get through it. So they're going to be on the inside or the outside. Whereas the integral proteins, since they're hydrophobic, they're going to get through because the middle part of this is hydrophobic. Right?
Yeah.
So, for instance, remember when I told you about the mRNA vaccines? Why do they use lipid nanoparticles?
So you can get through the hydrophilic layer.
The hydrophobic
Yeah.
part? Yeah. So they
Hydrophobic
can
layer,
go
yeah.
through, yeah. This is what it looks like right here. Okay. Let's see. So yeah, a cell membrane consists of a lipid bilayer with embedded proteins. So lipid bilayer here, and these are the proteins. So now we're going to talk about the chemical composition of the membranes. And this is a protein lipid ratio. Now, for the myelin sheath of the nerve, if the protein lipid ratio is 0.23, is there more protein or is there more lipid?
Wait, can you say that one more time?
Yeah. So for the myelin sheath over here, we have a protein lipid ratio of 0.23. Now, does that mean that there's more protein or more lipid?
Should be. Probably more lipid.
Why?
Well, it says approximate percent by weight is 79.
Just from the protein lipid ratio, though.
It's quite a low ratio.
Exactly. So essentially, how ratios work is if there's more protein than lipid, aka if the numerator is greater than the denominator, you're going to get a ratio that's greater than 1.
Yeah.
If you have a greater denominator than the numerator, you're going to get a number that's less than 1. What if there's equal amounts of protein and lipids? What would the ratio be?
If there was a what?
equal amounts of protein and lipid, what would the protein lipid ratio be?
0.50.
If there's equal amounts?
Oh, 1.
Good. So that's an important lesson in terms of ratios to always consider, because I see it come up on the MCAT all the time, and I always have to teach my students that. In terms of a ratio, you're just looking at if it's greater than 1, equal to 1, or less than 1.
Yeah.
All right? So yeah, since the protein lipid ratio is less than 1, it's 0.23, the denominator is greater, which means that there's more lipid than there is protein. So that's in the myelin sheath of a nerve. And it works as insulation.
Yeah.
All right, so now these are the 3 major classes of lipids in the plasma membrane. And again, so remember how it's a fluid mosaic model? So the fluid refers to the lipids, the mosaic refers to the proteins.
Yeah.
Got it?
Uh-huh.
Mosaics, they come from those like Roman art. Have you ever seen them? Like, they look like this right here.
Yeah.
Where they use tiny little tiles, right?
Yes.
So that's why they call it the fluid mosaic model, because the proteins are embedded like those tiny little tiles.
Yeah. So wait.
Yep.
Membrane. How can I put that in a sentence to make sure?
What?
Remember that? The fluid mosaic model.
Oh.
Membrane structure is pictured in a fluid mosaic model.
Oh yeah, the fluid refers to the lipids, and the mosaic refers to the proteins.
Lipids are the fluid mosaic. Got it. Okay.
All right. And it's funny, with one of my students, I also went over this word, amphipathic. And, I use that as an exercise to show that you don't need to know it to figure it out. What do you think amphi means? Like, think of words that start with amphi.
Amphibian.
Like, exactly. And what's an amphibian?
They're they can live in water and dry.
Exactly. Yeah. So amphi, it means 2. So amphipathic means that it has hydrophobic qualities as well as hydrophilic qualities.
Yeah.
All right, so we're going, since this class has a biochem like focus to it, and since biochem is going to be something you're going to take eventually, as well as organic chem, let's start getting used to drawing out the molecular structures, because that's something that you can't get away with by just typing or looking at notes. You have to draw it for you to gain like that muscle memory. Okay?
Okay.
So you heard what I said about the drawing part?
Yeah.
Okay. So, the lipid portion involves these 3 different types of lipids. And, here's like a drawing of it. So a phospholipid, for instance, has these hydrocarbon tails, which are essentially the phobic parts. And we have a phosphate group here and some type of amino acid that's going to be polar. So in this case, it's serine. For the phospholipid, we have cholesterol here, which is it's mostly hydrophobic, except for that OH part. So that's going to be the hydrophilic head. And we have a glycolipid as well.
Got it.
So what do you think a glycolipid would be?
What, based on the picture?
Just based on the name.
A lipid with a sugar attached to it.
Good. So in this case, we have galactose cerebroside, and we have a galactose over there.
Yeah.
Do you remember what lactose was?
Galactose?
Just lactose?
Oh, yeah, it's isn't that what like galactose derives from? Isn't that the monomer?
Okay. Along with
the polymer being the galactose.
So lactose is a disaccharide of which two monosaccharides?
Oh, when sugar. I know. Sucrose and galactose.
So galactose.
Glucose and galactose.
What's that?
Glucose and galactose.
Yeah. What? Since you mentioned sucrose, what's sucrose?
Sucrose is the disaccharide
Of
glucose.
And fructose.
And fructose, yeah.
Okay. Okay, so phosphoglycerides, right? So this is the category of phospholipids. There's various types. But this would be a good thing to kind of draw because it's kind of like a skim. It's kind of like just using boxes. So you don't have to draw the entire kind of structures of these. But it would be good to draw, glycerol, which is attached to two fatty acids. And, in order for it to be a phospholipid, instead of a, I guess, like triglyceride or whatever, it's not going to have three fatty acids, it's just going to have two, and instead of the third fatty acid, it's going to have a phosphate. And that can be attached to like a choline or a serine or something, whatever. As long as that phosphate's there, it's going to be a phospholipid.
As long as the phosphate is attached to the glycerol with the two fatty acids, it's going to be a lipid.
It's going to be a phospholipid.
Phospholipid, yes.
If it was a glycerol attached to three fatty acids, it would be a what?
Triglycerol.
Good. So, yeah. And if it was a triglycerol, it would be completely hydrophobic.
Hydrophobic.
Yeah, good. The phosphate introduces that polar aspect to it.
Yeah, so it can become amphipactic.
Perfect. Good. You're bringing all this together now.
Yeah.
What part of why is DNA negatively charged?
Because of the phosphate group.
Exactly. So when DNA wraps around histones, It means histones have what charge?
Um,
Like,
Positive, yeah.
yeah, good. All right, good. All right. So, all right, sphingolipids.
Wait, what's PC, PE, and PS?
Oh, so in the category of phospholipids, right? So I just kind of like said that as long as it's attached to a phosphate, a glycerol attached to two fatty acids and a phosphate, it'll be a phospholipid. But that phosphate could be attached to several other things. So the one that we have shown is choline. There can also be ethanolamine attached, as well as serine. So those are just the most common ones. So PC means phosphatidylcholine, PE means phosphatidyl-ethanolamine, and PS means phosphatidylserine. Those are the most common phospholipids.
Okay.
In the slide right before this one, you see the example of phosphatidylserine.
Yeah. Okay.
Yeah. So those are just
What are the names again?
the most common ones.
Yeah.
So in that slide, like see this over here, the first three over here. The first three are the most common ones.
Oh, okay.
So phosphatidylcholine, ethanolamine, and serine.
All right. I'm ready.
Okay. The next one is a sphingolipid.
Okay.
And so they're essentially derivatives of something called a sphingosine, which is, they described it as an amino alcohol with a long hydrocarbon chain. That doesn't really help us that much, but you can kind of see it in this picture over here.
Mm-hmm.
So this over here is the Sphingomyelin Backbone. So this is essentially the sphingosine aspect of it. See how it has this long hydrocarbon chain?
Yeah.
Yeah. You see this a lot in the brain actually. And it's interesting that like Ashkenazi Jews have lots of they have kind of two like things like genetically, like one it's like kind of they have like higher like IQs, but at the same time they have lots of neurological diseases, and they're both linked to like a sphingolipid a greater amount of sphingolipids in their brain. So I mean that's just what I think of when I see them. But yeah, it's another type of membrane lipid, and sphingomyelin is a specific type that's especially in the brain.
Got it. Okay.
Okay. And the third type is cholesterol.
So wait, what should I put down for sphingolipids then?
So for each of these, I want you to like draw the structures. Like you can just draw like this type, like you know where it says choline, like in the boxes.
Yeah, I have that.
Okay. And you know, if you want, you can draw like the structure that's like here, but it might take a little longer. But it's definitely worth it. Like, you know, maybe not during the session, but like when you study on your own to be able to draw the structures and get that muscle memory for it. Because with like chemical structures, it's like math where you can't like cram for it. You need the muscle memory of like doing it, actually writing it out and stuff.
Yeah.
So, like if I can describe a phospholipid, it would be like with a glycerol backbone, and a sphingolipid would just have a sphingosine backbone.
Okay, so sphingolipids have a sphingosine
backbone, yeah. And you know, if you want, you could say the sphingosine is essentially just it's an amino alcohol with a long hydrocarbon chain.
alcohol with a
long hydrocarbon chain. So that kind of is why it has this vertical region to it.
Yeah.
Like right here, do you see that?
Yeah.
So like compared to this, so glycerol, right? In a phospholipid, glycerol has the two vertical regions as fatty acid because of the long, you know, tails. Sphingosine has its own vertical region, which is its own hydrocarbon tail.
Got it.
All right. And then there's cholesterol. So cholesterol, you know, essentially has this rigid planar steroid ring structure. And it'll have a nonpolar hydrocarbon tail along with a polar head group. And it's going to be pretty much embedded into the phospholipid bilayer, and it affects how rigid that bilayer is. So it can constitute 50% of animal cell plasma membranes. It's not in plants or bacteria. I know bacteria has, I think, like alpha or tocopherol or something else. Teichanoic acid. You don't need to write that down, but bacteria has that instead of cholesterol. But animal cells have cholesterol. They're much smaller. They're pretty much tiny compared to, you know, phospholipids and sphingolipids. And they're less amphipathic. And they just interfere with the packing of the phospholipids. So at lower temperatures, it makes that membrane more fluid, and at higher temperatures, it makes it less fluid.
it's important to note that they're absent from plants and bacteria, yeah?
Yeah.
And what else should I note from here?
Oh, just how it affects the fluidity of the membrane at the different temperatures.
Oh, okay. Okay.
Okay. Yeah, the next couple of slides just talk about how in water essentially a lipid bilayer self-assembles. It forms naturally. And that's just due to like what's energetically favorable.
It happens naturally, right? That's what you said.
Yeah, based on what's energetically favorable. So this diagram over here is pretty good. So, you know, like it'll essentially just like from this position where it's energetically unfavorable, the hydrophobic tails are the white layer and the red layers are hydrophilic. And if it was just left like this as this sheet, the white regions are exposed to water at these edges. See over here, there's an edge there and there.
Mm-hmm.
Like here, here, and there. So in order to like not let those hydrophobic edges exposed to water, it'll close in on itself, forming this sphere, which is a liposome.
I see.
Yeah. That's like, you know, why like oil in water will form like a sphere as well, or like a circle. And it, you know, even if you take away the hydrophobic, hydrophilic parts of it, like take a liquid in space, what shape is it going to be? Same thing with rocks in space that make up a planet. What shape is that? Or gases in space that make up a star. What shape is that? They're all one shape, right? A sphere.
Mm-hmm.
Because that's the most energetically favorable shape that something can be. Because it has the lowest surface area.
Yeah.
All right. So now, the nature and importance of a lipid bilayer. Essentially, what does it do? So these are the three things that it does. One, it determines the physical state of the membrane. The lipid composition, that is. Two, it influences activities of some of the membrane proteins. And three, it may provide precursors or chemical messengers. Yeah, look at this. Do you see?
What is that?
My cat that I just adopted.
Oh.
Yep.
Did you just get her?
Mm-hmm.
Or him?
Yep.
Nice. Okay.
Look at this. Look at this. Come here. Oh, whatever. Anyway. All right. Phospholipids, glycolipids are distributed asymmetrically in the plasma membrane.
Okay.
So yeah, you should write that in the plasma membrane, glycolipids and phospholipids are distributed asymmetrically.
Yeah.
However, in the endoplasmic reticulum, they are they're randomly distributed. And that's done by scramblases. They randomly transfer some phospholipids to the inner leaflet. Oh, and newly synthesized ones are added to the cytosolic membrane.
Say that one more time?
So for the endoplasmic reticulum, as new lipids are synthesized, they're added to the cytosolic side of that lipid bilayer, the one that's facing the cytoplasm. And an enzyme called scramblase transfers some of those random phospholipids that are on the cytoplasmic side and moves them to the inner part, which is called the lumen.
What was the lumen? What?
The lumen is a general term that means it's the inner portion of a membrane of an organelle that has a membrane.
Got it.
So here's like an example over here. So here we have the lipid bilayer of the endoplasmic reticulum. Cytosol is on this end. The lumen is on this end. When new phospholipids are made, they're added on the cytosolic part, right? But scramblase transfers some of them from the cytosolic part to the one that faces the lumen. And the end result is that there's symmetric growth of both ends. Now, this is unlike the plasma membrane, where it's asymmetrically distributed. In the endoplasmic reticulum, it's symmetrically distributed.
Huh.
All right? So you got that?
Yeah.
Okay. Now there's flipases and flopases, and they also work to... So unlike scramblases, which work to make it from asymmetric to symmetric, flipases and flopases work to maintain and to make an asymmetric distribution. And you can see them in the plasma membrane. You can also see them in the Golgi apparatus.
Okay.
So here is an image of the lipid bilayer of the Golgi apparatus, where the cytosol is on the bottom this time, and the Golgi's lumen is on the top. And what essentially happens is that a new membrane from the endoplasmic reticulum is added to the Golgi. Look at this now.
Didn't like that. I'm ready.
All right, so do you get this about in the Golgi? So essentially, in this picture, the Golgi lumen is on the top, on like here. Cytosol is here. Delivery of a new membrane from the endoplasmic reticulum comes in, and the flipase catalyzes the transfer of specific rather than random phospholipids to the cytosolic layer. So essentially, they move specific phospholipids, for example, phosphoserins, to the cytosolic part. And flopases move specific phospholipids like phosphodietylenines to the outer leaflet. But the main thing to take away here is that it makes the distribution asymmetric.
Got it.
Alright, some functions of membrane phospholipids and cell signaling. Okay, let's see. Extracellular signal, activated receptor protein causes this. Phosphorylated inositol phospholipids. So it causes the phospholipid to phosphorylate and send a relay signal. Okay. This one, okay. That's just an example of whatever. The cell surface is covered with carbohydrates. Okay. Which are linked to the lipids and proteins, aka that's how you get the glycolipids and glycoproteins. 2 to 10% plasma membrane by weight. Less than 10% in glycolipids, greater than 90% in glycoproteins. So here are just examples, I suppose. Alright, now structure and function of membrane proteins. All right, so here's the image of the fluid mosaic model again. So now we're going to talk about the functions. So there's going to be transporters, anchors, receptors, and enzymes.
Okay.
Example of a transporter is a sodium pump, which actively pumps sodium out of cells and potassium in cells. That's how neurons work. Now, and then there are anchors, an example of which are integrins, and they link intracellular actin filaments to extracellular matrix proteins. Example of that is like in muscle cells, myosin, actin, filament, that kind of thing.
Yeah.
Of course, there's receptors. So an example they're using is platelet-derived growth factor receptor, which binds extracellular platelet-derived growth factor and as a consequence generates intracellular signals that cause the cell to grow and divide. And then, of course, enzymes. Adenylyl cyclase is the example that they're using, and that catalyzes the production of intracellular signaling molecule cyclic AMP in response to extracellular signals. Any questions there?
No. I guess I can just basically copy and paste the table and stuff like that. That works.
Yeah, but I always recommend writing it out because it just stays with your memory a lot longer because it's like muscle memory.
All right. So I was gonna say.
But you don't have to write it out like during our session. You can do it like when you're studying like outside.
Yeah.
But I would definitely recommend that you like handwrite them instead of copying and pasting it because otherwise you're it's easier to forget. And I don't want you to forget things like 5 cap and things like that.
All right. What's next?
Okay, so see this over here, this slide?
Mm-hmm.
So the larger categories of integral proteins and peripheral proteins have subcategories. So tell me again what integral protein is and what a peripheral protein is.
Integral protein and a peripheral one?
Yeah.
Integrals are hydrophobic, and peripherals are hydrophilic.
All right, but tell me, like, why? Like, what?
Well, the integrals embed themselves in the bilayer and don't go out or in.
Good.
And then the peripherals embed themselves out and in.
Good.
But not, no.
So now, within the category of integral membrane proteins, there are subcategories. So there's the transmembrane ones that go completely through. Okay? They completely go through the membrane. But there's also a monolayer one, which doesn't go through the entire bilayer. It just is attached to one layer. And then there's a type that is called lipid linked, which is just attached to a lipid, which is attached to, which is in that bilayer. So not all of them are the transmembrane ones.
Okay.
So these are the transmembrane ones, and as you can see, they're completely within, they completely go through the bilayer. But there's also a monolayer one, which just goes through one layer. And then there's a lipid-linked one, where it essentially looks like a peripheral one, because the protein part is just attached to a lipid, which is attached, which is in that bilayer.
All right. Wait, you're breaking off. Can you say what lipid linked is again?
Sure. So the lipid-linked ones essentially look the same as a peripheral one. The only difference is that they are attached. They look like a peripheral one because the protein part is completely outside the bilayer. But they're still considered integral because they're attached to a lipid, which is in that bilayer. So it's just these lipid parts, these zigzag lines over here, that are part of the bilayer. The protein is outside of the bilayer, so it pretty much looks like a peripheral one, but it's technically integral because it is anchored by that lipid.
Okay.
So those are the three subcategories of integral membrane proteins. The peripheral membrane protein doesn't really have other subcategories. It just it's just a
it is.
What's that?
It just is.
It is. I would say it's essentially the same as a lipid-linked one, except it's attached or anchored by a protein.
Yeah.
So that is kind of weird because why, lipid-linked, the protein is completely outside. Why is it called integral? And it's just because it's linked to a lipid that's in the bilayer. And for protein-attached ones, you don't count the part that's you don't count the protein that it's attached to. That would be separate. So in actuality, if you look at something like this, it actually includes a peripheral protein in addition to a transmembrane protein.
All right, say it one more time.
So take a look at this over here.
Yeah.
So this protein-attached peripheral protein has the peripheral part outside, but it actually has a transmembrane protein as well. Because this is identical to this over here.
Got it.
See this part, this gray part over here?
Yeah.
This gray part is the same as this over here. This
Okay.
over here. See that?
Yeah.
So that's why we call it a peripheral protein, because the protein is attached to a transmembrane integral protein. So that's why we don't call it. That's why, you know, a lipid anchored one and a protein anchored one are not both integral, because the protein anchored one, the anchor that is the protein, is integral.
Okay.
Now, why don't you tell me what this kind of thing is called right here?
Coiled protein?
What's that?
A coiled protein?
Kinda. It's a type of secondary structure.
Oh,
What about
oh,
this?
hydrogen one?
Huh?
No. Sorry.
I mean, like, it is caused by hydrogen bonds between the amine nitrogen and the carbonyl of the carboxyl group in the amino acids, but it's called an alpha helix.
Yeah, there we go. Okay.
What about this?
I remember primary was, yeah, amino acids, and tertiary was like the 3D model.
Mm-hmm. What about these?
Beta helices?
So, a helix has the coils, right?
Yeah.
But these are flat.
Oh.
So they're beta pleated sheets.
Oh, that, okay. Got it.
Yeah. And they have different directions, so that's why there's the arrows.
Okay.
Yeah. And they're anti-parallel.
Got it. Okay.
Now, the transmembrane ones, or integral proteins, right? This region is hydrophobic, but the regions that are outside, right, are hydrophilic, right?
Yes.
Like, look at where my cursor is over here. So this part is hydrophobic, but this
Okay.
part and this part are hydrophilic, right?
Okay.
Right?
Yes.
So that makes them amphipathic.
Okay.
All right?
Okay. What's next?
Yeah, so here's another like drawing of a transmembrane, integral protein, and you can see the alpha helix over here.
Yes.
All right. Okay, now not all transmembrane segments are entirely hydrophobic.
Wait, one question.
Yeah.
What does it mean by required detergents to solubilize?
Oh yeah. Okay, so I was gonna talk about that after. So basically, a detergent, kind of like, when you use it for laundry and stuff, it's something that breaks apart, like oils and fats and hydrophobic things like that. So if you want to, first of all, like, how do we know that there are transmembrane proteins and things like that? We take our cell and we use detergents to break it apart. There's different ones. There's like SDS, sodium decyl sulfate, I think, or something like that. In our bodies, bile is a detergent. It breaks apart oils and fats and turns them into smaller kind of pieces, that smaller micelles or circles that can be readily digested and stuff. So to separate these separate the membrane proteins from the membrane, we use detergents to do that.
Okay.
Yeah, they'll talk more about it in some of the later slides. So yeah, and then here's just a closer kind of drawing of like an example of the alpha helix in a transmembrane protein.
Yeah.
All right, but not all the transmembrane segments are entirely hydrophobic. So for example, if it was an ion channel, right, it's going to have to permit the flow of ions. And ions are, by nature, hydrophilic since they have a charge. So here's an example of, 4 alpha helices, kind of like around each other, and the parts that are close to this center area are the polar parts, the hydrophilic parts.
Yeah.
Because these things are really just the side chains. So the side chains that are polar, aka polar amino acids, make this polar core, which enables the transport of ions into and out of the cell.
Got it. Okay.
Okay, and an example of it is bacteria rhodopsin over here. So, we see these transmembrane alpha helices, and then this center region over here is where the polar side groups are, so it enables the transport of, these protons, the hydrogen plus ions.
See. Okay.
So yeah, this example, bacteria rhodopsin, is an example of the above, where you have these, transmembrane proteins, alpha helices, but the side groups or side chains that are situated in the center are hydrophilic, and that enables the transport of ions. So you have this, H plus ion being able to be transported from the cytosol to the extracellular space, aka outside of the cell, through this, ion channel.
Got it.
All right, now methods. All right, tell me when you're ready.
I'm ready.
Okay, methods to use to understand integral membrane proteins. So this is when we're going to start to enable. In order to understand it, we're going to have to break it apart, right? Red blood cell plasma membrane disruption with detergent. So here are examples of detergents. This is the one that I mentioned, sodium dodecyl sulfate. It looks like this.
Uh-huh.
So this is the polar region. This is the non-polar region. There's Triton X100. So, they're going to be amphipathic. Like, any soap is going to be, like, amphipathic as well. Because there's going to have to be a non-polar part that connects to, like, the oils and stuff and the fats on your body. And the polar part that the water attaches to when you take a shower so it can, get rid of it all. These detergents can substitute for phospholipids and make hydrophobic proteins soluble in water. So in order to study the structure and properties of membrane proteins, we solubilize it with detergents. So here we have, the lipid bilayer and a membrane protein, right? There's two alpha helices here. And we want to study that membrane protein. So we need to have it removed from the lipid bilayer. So we add a non-ionic detergent. So I guess the difference between SDS and Triton X100 is that SDS is ionic. It has a negative, ion right there. And Triton doesn't have ions. So we add something like Triton, non-ionic, and essentially we solubilize this membrane protein in water. So now it's in an aqueous solution, and you see how the parts that would normally be touching the hydrophobic parts is now just attached to the detergent instead. So now we have a detergent solubilized protein. So essentially, they work to stabilize the integral protein in an aqueous environment. And now we can analyze it.
I see, okay. So it breaks them apart. I got it.
Mm-hmm.
It makes it all aqueous. So it can be
so
on the same realm.
Yeah, the outer parts of the alpha helices are still hydrophobic. It doesn't change that. It's just that the detergent is also, that part of the detergent that it's attached to, the tail, is also hydrophobic. So it kind of shields it when it's put into an aqueous solution.
Okay.
All right, next slide says identifying transmembrane domains, and they're usually hydrophobic alpha helices. So let's see. So we have this one over here, glycophorin A. It's hydrophobic amino acids. Okay. Let's see. Around 20 amino acids are needed to span the membrane. And then they do the hydropathy plot, which is testing its aversion to water, how much it dislikes water. And you remember, like, this delta G thing, Gibbs free energy?
Yeah.
So, like, delta G should be negative if it is, energetically favorable. It'll be positive if it's not. So when they put it in water, right, that's how they can tell, like, which amino acid residues are hydrophobic and which are hydrophilic. Because if it's in water and its Gibbs free energy is positive, which means that it's not energetically favorable, then that means that it has that region is hydrophobic.
I see. Just quickly with the helices part. So you, just to make sure that what you said I have correct. You said that the outside part of the helices stays hydrophobic. It's just the inside that becomes hydrophilic.
Oh, no. So the outside remains hydrophobic, but when it's in a membrane, the membrane is also hydrophobic. Now, when you take it out of the membrane and put it in something hydrophilic like water, you need the detergent to be around the hydrophobic part. So in this picture over
so
here,
how can I write that down? Because it's a little confusing.
Oh, let's see. Here, I'll like draw something. Okay, so let's see. This is a top-down view of the alpha helix. Let's see, I'll say green means hydrophobic. And, well, wait, I'll use different colors actually. Okay, so this is a top-down view of an alpha helix. So, the inside part is here. This and then these are the side chains that are on the outside, the R's. So they're hydrophobic side chains. So the outside of the helix is hydrophobic, right?
Yeah.
And this is it when it's inside of the plasma membrane. So the lipid bilayer. So that's also hydrophobic, right?
Yeah.
Okay. Now we're adding a detergent, which is this red thing with that tail. And the circle part of the red thing is hydrophilic. The tail is hydrophobic. Now, what happens is the detergent, the hydrophobic part, attaches to the hydrophobic side chain, right?
Yeah.
Leaving the hydrophilic part on the outside, these circles. So now you could put it in something like water, and it doesn't need to be embedded in that lipid bilayer anymore. Because, instead of the lipids surrounding the hydrophobic side chains, you have the hydrophobic part of the detergent that is surrounding the hydrophobic side chains. But you also have the hydrophilic part of the detergent on the outside. So overall, what you're getting here is here you have the outside of the alpha helix being hydrophobic and the environment being hydrophobic as well. But here, we're putting it in an environment which is water, which is hydrophilic, and we're not changing anything about the alpha helix. We just are putting a shield around it of that detergent, which has a hydrophobic part, which is around it, acting like a shield, and then a hydrophilic part, which enables it to be soluble in water.
Got it.
All right. Now, maybe another example could be just this is how a soap works. When you have imagine using taking a shower with just water, right? It's going to be the water is going to solubilize things on your body that are water soluble, but if you have
oils and stuff, it won't take out. So
exactly.
The oils basically become the detergent.
Yeah. So how do you get the oil out, right? So if you used oil to bathe, it's going to solubilize the oil, but it's not going to solubilize the water soluble stuff. So soap basically is a detergent. It has a hydrophobic part and a hydrophilic part. So the hydrophobic part will attach to the oils, and the hydrophilic part will solubilize in the water that you use to take a shower and pull that entire thing out. So it'll pull the hydrophobic part will be taken away with the water that you're using to shower, and the hydrophobic part will be attached to the oils, pulling them away.
Yeah.
So yeah. So essentially, we're taking something that has a hydrophobic outside, which is in a hydrophobic region like a lipid bilayer, and we want to put that into a water environment, which is hydrophilic, and we do that by adding a detergent, which just takes the hydrophobic part, doesn't change it, it just attaches to it, and it has its own hydrophilic part, which is what makes it soluble in water.
Okay.
So, they describe it by saying that the detergents just substitute the phospholipids in stabilizing the transmembrane proteins while also making it soluble in an aqueous solution. So this lets us now analyze that alpha helix, that transmembrane protein, like directly, because we can't really analyze it when it's part of the lipid bilayer, because we have the lipid bilayer to worry about. We have other transmembrane proteins and glycoproteins and all sorts of crap, so we need to isolate it.
Okay.
Yeah. Another example of a detergent in our bodies is bile. So like, let's say this is just, a bunch, it's like fat, right?
Yeah.
And we want to solubilize it, but to do that is, so bile basically turns the fat into much smaller little droplets of fat. And when it turns, what's easier to digest, this or these?
On the little ones?
Yeah, because there's more surface area. So there's more area for, enzymes that would digest it to be able to, come in and digest it. So bile is something in our bodies that acts as a detergent for fats. Here's another picture over here from this textbook that I have.
Mm-hmm.
So we have, transmembrane proteins, and then they add a detergent, and, it can form, these things called micelles.
Got it.
All right. Make sense?
Yeah. Let's see
Okay.
if we can try and finish this one up so we can do presentations on Monday and Tuesday next
Okay.
week for the test.
All right. Let's see, where were we?
Speed run what we can and see
Mhm.
what's more important.
Okay, so once you have the transmembrane protein out in water, you do a measure of its Gibbs free energy to see which parts are energetically favorable, which parts are energetically unfavorable. The ones that are energetically unfavorable will have to be hydrophobic, because now it's in water, right? It's an aqueous solution, aka hydrophilic, and that helps us identify those regions that are hydrophobic will have to have hydrophobic amino acids. So that's going to help us identify the amino acid content of the protein. which, you know, we're trying to do because we're trying to analyze the protein and identify what it's made of.
Yeah.
Okay. All right. So far we've just been talking about how to analyze the transmembrane, the integral proteins. Now for peripheral membrane proteins, it's a lot easier because they're not attached to the lipid bilayer. So you don't need a detergent. So, you know, they're essentially just attached through weak electrostatic bonds with lipids or integral membrane proteins. So they can be solubilized with aqueous salt solutions. You know, they could be enzymes, specialized coats, provide mechanical support like integrins, or transmit transmembrane signals. So that's, you know, how you can analyze peripheral membrane proteins. And there's lipid anchored membrane proteins.
Okay.
And they differ in the anchor that is used and on which side of the membrane it is, if it's on the outer side or the inner side. So, you know, the two types, there's GPI, which is glycophosphatidylanoxetol. Inositol. So its membrane side is on the external, the one that faces outside of the cell. And its anchor is a glycolipid. And then the other type is a fatty acid anchor, and that is on the internal side of the membrane, and its, you know, its anchor is obviously going to be the fatty acid. Examples of the first type are various receptors, enzymes, cell adhesion proteins, which makes sense because if it's for cell adhesion, like sticking to another cell, of course it's going to be on the outside, right? If it's a receptor, it's going to be on the outside, right? Now, for the ones that are facing the inside, it's just, the only example they gave is like a kinase, which is just something that transfers a phosphate from one thing to another thing. It phosphorylates things. I'll be right back. I'm just going to use the bathroom.
Okay.
Okay. All right. Next is membrane lipids and membrane fluidity. Let's see. Laser tweezers. Okay. Laser tweezers. Okay, I guess all you need to really know here is that, like, you know, the fluidity depends on temperature. So if it's colder, it is going to be more rigid. So it's like a gel, which is frozen-like. That's caused by the cholesterol. And as the temperature gets warmer, it becomes less rigid, so more like a liquid. Fluidity depends on chain length and degree of saturation. So, this is kind of like an example that they use with butter and olive oil. Butter doesn't have like double bonds in it, so it can be packed into like a solid, whereas olive oil has a double bond in it, which creates a kink, like a bend in its hydrocarbon tail, which makes it more difficult to pack, closely together, which is why it's a liquid at room temperature.
Okay.
And then I guess you can write that cholesterol disrupts the close packing of fatty acids and interferes with their mobility. So it creates this condition. So instead of there being these sharp differences in whether it's going to be like a solid or a liquid, it just creates like a condition of intermediate fluidity where it's not going to be completely liquid or completely solid. It'll be like in between, like a gel state and like a liquid state.
Got it.
Here's like a graph, like a graph to that kind of shows you. So the green one, this green line is butter, and you could see from a temperature of 37 degrees Celsius to a temperature of like 43 degrees Celsius, that narrow range, it goes from a solid to a liquid. Which means, it can change wildly just in that narrow temperature range. Then you have oleic acid, which is olive oil, in the blue curve. And you see that from like negative 23 to negative 10 degrees. It goes from solid to liquid as well. So that's also a very narrow temperature range. For cells, obviously you don't want it to be able to, melt or go from a solid to a liquid in such a narrow range, because then when the temperature changes, our cells are going to be like completely either frozen or liquid, which means our cells won't be very durable. It'll be at the mercy of the environment's temperature. So, cholesterol helps to make this intermediate kind of phase between solid and liquid. So you see that between like negative 23 degrees all the way to positive 20 degrees, it stays solid, which is a huge temperature range. So that's, the benefit of cholesterol in our cell membranes, because without it, we would have tremendous changes in phases from solid to liquid in a very narrow temperature range. Cholesterol helps make that range really, really wide, so the cell can, do its thing regardless of the temperature. So that makes sense?
Yeah.
All right. Maintaining membrane fluidity. Internal temperature of most organisms, not birds or mammals. So I guess this is like cold-blooded animals. changes with outside temperature. Organisms respond by altering membrane lipid composition. So there's enzyme types called desaturase, which takes single bonds and turns it into double bonds. And double bond means that there's a kink created, which means it's harder to compact it into a solid. So, if the temperature drops and your cells want to freeze, desaturase makes those single bonds double bonds, which makes it harder to freeze. So yeah, that's why it says at lower temps. And phospholipases and acetyltransferases reshuffle fatty acid chains to create lipids with two unsaturated chains. I'll be right back. Any questions so far?
No, it's pretty self-explanatory.
All right.
Not bad. Okay.
These are just talking more about the asymmetry of the type of membrane lipids. So, the cell membrane is asymmetric. Endoplasmic reticulum is symmetric. Golgi is asymmetric as well. This is like an image of it. And then there's lipid rafts, okay. Alright, now dynamics of the plasma membrane. So possible movements of phospholipids in a membrane. Okay, so there's transverse and lateral. So transverse is when it moves from one side to the other. So, from the inner portion to the outer portion, or vice versa. Lateral shifts is just shifting like sideways, but on the same side. So it'll stay on the inner side, it'll just move from one point to another. Lateral shifts obviously are a lot easier. They happen like in a millionth of a second. Transverse ones are like the flip-flops, a lot harder, so it happens once every 28 hours. And flipase and scramblases can also help them do that. And there's also flex, which is just when the one just kind of twists.
Just quick going back to that slides about cholesterol.
Yep.
What do you call it? Those three things, right?
Oh, which ones?
The 3 bullet points there? Going up, going up.
Oh, this, yeah.
That all has to do with cholesterol, yeah?
Yeah.
Okay. Making sure.
Yeah. So the first
Okay.
bullet point is like, you see in these pictures, it it disrupts the close packing of the fatty acids, so it interferes with their mobility. They're not able to move as much.
Yeah, but it creates like an intermediate space of fluidity, so it's like neither liquid nor solid.
Yeah. This graph really shows that well.
Yeah, the drawings don't really do much. Okay. I'm just making sure I have that. Okay. And can you explain lipid rafts really quick?
Oh yeah. So
again.
Oh yeah. So okay. There's asymmetry in the types of lipids on the cell membrane, meaning that there's going to be more like phosphatidylcholine on the outer portion and more like phosphatidylethylenolamin on the inner portion. And so this image over here kind of shows it. Like this is kind of like on one side of the membrane, and we have basic lipid bilayer over here, and the rafts are just like these parts that kind of like come up a little bit more. So like we have here sphingomyelin. So okay, like here it's like the analogy could be like the ocean is this stuff, which is PC. It's phosphatidylcholine, and the raised portions are the rafts. So the sphingomyelin over here, this part is not just raised; it like has like a peak, like this tip. That's GPI glycophosphatidylcholine inositol, and here's like a side view of it. So we have the regular ones, which are just the PC. They don't really come above as much, and then we have like GPI, which comes up all the way to make this peak, this like spike over here. Yeah. So lipid rafts are just called that because they look like rafts, sort of, because they're raised compared to like the regular phosphatidylcholine which is kind of like the analogous for like the sea or whatever, the water part. So they're just, it's just to show you the asymmetry of the membrane lipids. Like, see over here, on the exoplasmic side, the outside, there's a lot of PC.
Yeah.
So that's why most of this is PC. And then some of it is PE, but less of it, and there's also some SM over here, which is this stuff. So it looks like...
PC is like a glycolipid, yeah?
It's just phosphatidylcholine. It's a type of phospholipid.
Okay.
So, like, if you think about it, right? Like, look,
it's not a glycolipid.
Correct. So, like, glycolipids. So, in this diagram over here, right? We have so these are just the regular phospholipids. This could be like a sphingomyelin or something. So it's raised a little bit. These things in between are cholesterol, and then these things are the sugars, because sugars are always like, a hexagon or like a pentagon, like, like pentose sugar or like things like that. So these are the glycolipids. So they're raised. See how they're raised? So that's what you see here, where they go up. They're raised from the surface, from the flat surface.
I'm just asking that because, like, 2 slides before, it has PC as, like, I think a glycolipid? No, yeah, the chart you just passed. There. No. Down. No, I mean, no, sorry. Yeah, there.
Here?
No. The side of go up. No, like down. The asymmetry of membrane limit?
Oh, yeah. Uh-huh.
Yeah. Why does it have PC as like the one on the outside if it's not a glycolipid on the outer leaflet?
PC, that's this over here?
Yeah.
Yeah. What were you saying?
It has that as, it says glycolipids outer leaflet.
This is just, all right, so this is just saying that there's asymmetry.
Yeah.
In this chart over here, it doesn't have anything about glycolipids, but this just means that if there are glycolipids, they will be only on the outer portion, on the outer leaflet. So in this diagram here, you can see them with as being these things, because they have basically the universal kind of symbol of a sugar is like a hexagon or something, because that's the shape of like sugars.
Yeah.
So that's what these blue hexagons are. But in this graph over here, it's only dealing with phospholipids. So that's why these arrows are not pointing to any of the ones with the blue hexagons.
Okay. And then Lipid Wraps. Okay, that was pretty easy to understand.
Yeah, do you
The
see
PC
how
the PC is like the one that's all the way down,
yeah, kind of like
like
the
the gray
sea.
ones, basically.
Yeah, it's kind of like the sea or like the ocean, and the raised ones are like the rafts floating on the ocean.
Yeah.
And do you see how it corresponds with the picture on the right of it? That's like a side view of it, and
Yeah.
the you see how like it rises, like it rises when you have these red hexagons, which are the sugars, and it rises when you have the the blue ones, which are just the sphingomyelin, and then it rises the highest when it's GPI. And that's why on the left image, the GPI are those spikes.
I see.
I have like another kind of image of it here from this textbook.
Yeah.
So
I just want to clarify. The
Yeah.
PC is the gray, right?
yeah,
And then the blue is the SM, and then the GPI is the red with the brown.
yeah,
Got it. Okay.
yeah. And the they don't have this labeled, but the spikes that are smaller are the ones that are just with the red hexagons, but without the GPI.
Okay.
And the thing like this, so this is like
Wait, what was with the little hexagons?
oh, these are just the smaller spikes.
Yeah, the GPI.
Oh, so the GPI is the
Just
really
the protein.
the big spikes, but
Uh-huh.
and they just don't have this labeled, but these smaller spikes are like this right here,
Yeah.
which are just these sugars.
Oh, just sugars and not SM, PC, or GPI.
Yeah, the GPI would be attached to like the sugar. So like here's like another image over here on this over here. So you can kind of see it's like a side view, right? So PC, it's like 3.5 nanometers. Like that's how thick it is. Now
Yeah.
PC with cholesterol, it gets a little thicker. Now SM, it gets more thicker. So so we can think of the PC as just being like the sea level, and then the first raft is SM. So you see like how it's thicker here, so it rises a bit. That's why it appears to be like a raft.
Okay.
So yeah, so it makes sense that like this image on the right is like a side view of the lipid raft thing on the left, right?
Yeah.
Okay.
Okay, and then this chart with the dynamics of the PM. It's pretty easy to understand.
Oh yeah, that's like the transverse one once every 28 hours. Lateral ones are on the same side, which are easy to do, so it takes a millionth of a second to do. The flex is just like, you know, if it twists.
Yeah.
And yeah, do you want to leave it at this and pick up next time? Or are we?
No, I still got a little bit to go.
Okay, let's finish it. Okay. Protein.
Well, no, it's going to be a little bit to go, and if you got another student, it'd probably be better we just finish Monday quickly, and then we go on to the next one.
I don't have another student. But the rest of the stuff, yeah, it's up to you, because the frap thing might take a bit.
Yeah. All right, so we can wait until Monday.
Okay. All right. So, yeah, so like go over this, you know, go over the notes and stuff yourself. And, you know, when we meet on Monday, I'm going to ask you like some questions, you know, before we start. So be ready for that.
Okay.
And, I don't know, do you want me to send you this textbook? Because it has, like, this is like a good chapter on lipid bilayer, if you want. I don't know.
Yeah, no, you can.
Okay.
Send it.
Yeah, it's like it's a good one. Okay, so I'll send that to you, and yeah, just you know, make sure you study on your own time as well. And when we meet next time, I'll ask you some questions first, and then we'll continue where we left off here in these slides. Okay. All right. So that's
All right,
it.
then.
All right. Take care, man. Feel
Take
better,
care.
hopefully. All right.
Yeah.
See you, man.
See you.